So as we've been talking about various protein structures, primary, secondary, tertiary, now we are at quaternary structures. And if we can understand tertiary structures, we can understand quaternary structures because the same rules apply. So the main driving force of quaternary structures is once again the hydrophobic effect, where the hydrophobic non-polar molecules want to cluster on the inside, and the hydrophilic polar molecules will cluster on the outside because this is more energetically favorable rather than the hydrophobic groups interacting with the water. They do not like interacting with the water. Now, we can also have disulfide bonds, and remember that a disulfide bond is essentially two cysteine amino acid residues that are bound together like this. Now, what's important to note is that cysteine has a pKa of about 8.3. So when the pH is above 8.3, our sulfurs are deprotonated and we're able to get this disulfide bond. But if the pH falls below 8.3, then our sulfurs will be protonated and we will not be able to form this disulfide bond. And this is very important because if, for example, our quaternary structure has a disulfide bond, at low pH values, that disulfide bond will be disrupted, which will overall impact and disrupt our quaternary structure. Okay, so the next factor is hydrogen bonding. And remember that we see hydrogen bonding in secondary structures as well. But in secondary structures, when we talk about hydrogen bonding, we are talking about the peptide bond oxygen and nitrogen having partial negative and partial positive charges that interact with each other via hydrogen bonding. But in quaternary structures, we see that hydrogen bonding on those peptide bonds, but we also see hydrogen bonding between different R groups of amino acids. In secondary structures, our R groups do not interact, but in tertiary and quaternary structures, our R groups do interact via hydrogen bonding. Now, the fourth driving force or stability factor of quaternary structures is electrostatic interactions. Now, we can imagine that on our quaternary structure over here, we have two subunits. And let's say that one subunit had an aspartate, okay? We know that, for example, let's say that before we had two tertiary structures. One of this was this white one and one of this was this purple. So I can draw that out. Let's say we had this tertiary structure over here, and I'll draw it in blue since we can't use white. But we have these two tertiary structures. And now let's say we know that the exterior is most likely hydrophilic, and the amino acids that have to be on the outside must be hydrophilic. So let's say I have an aspartate over here. If this aspartate is negatively charged, and this tertiary structure over here, for example, has lysine. We know lysine has a positive charge. It's also hydrophilic. So this positive of lysine and this positive of aspartate, negative of aspartate, positive of lysine, they'll be able to interact with one another. I'll draw this out to make it more clear. So let's say we have a negative on the outside over here and a positive. Now these two tertiary structures are able to interact with one another. And if we have a bunch of interactions between these two tertiary structures, they come together and we get the quaternary structure. So a quaternary structure is essentially we have these tertiary structures that are already made and we're bringing them together. We're bringing them together to form these quaternary structures. And we can do this through electrostatic interactions or even hydrogen bonding. Now, the last thing that affects quaternary structures is metals, and we discussed this before. Now, we can remember that, for example, if we have an oxygen, for example, glutamate, it has an oxygen that has a negative charge at high pKa values. And if we want to balance that charge, we can use we can use metals that have a +2 charge. For example, magnesium has a +2 charge, and that can be used to balance out the negative charge of the oxygen. Now, if we don't have metals to balance out that charge, and we have a lot of groups that have a negative charge, we will see repulsion, which is not beneficial to the quaternary structure. It's actually going to disrupt it. So, out of these 5 characteristics of quaternary structures, these structures can be disrupted if we have too many negative charges or if we have too many positive charges and there's repulsion. At high pH values, we will see less hydrogens bound because it'll be deprotonated, so we'll see less hydrogen bonding on the R groups. But remember, there will still be hydrogen bondings on the backbone of the peptide because those are not ionizable groups. And then if our pH alters, it's going to affect our disulfide bonds as well, because we know that cysteine has a pK of 8.3, it's ionizable, it has the ability to be protonated or deprotonated. So quaternary structures, these are the stabilizing factors, but also the factors that if they don't go well, or how they're supposed to, by altering pH or temperature, we will see denaturation of the quaternary structure. Now, this is just a diagram of how we get to our quaternary structure. We have that primary amino acid sequence. We get that secondary structure with the alpha helix, the beta-plated sheets. And then when we see R group interactions, we get our tertiary structure. And then we have several tertiary structures, we get our quaternary structure. And that is essentially quaternary structures of peptides. What makes it different from secondary structures is that hydrogen bonding is able to happen with R groups in quaternary and tertiary structures.